Well, I've sat through lots of meetings like this and lots of summary talks, and I, I'm guessing most of you went to college and really understood these talks, and I'm not going to repeat what you've heard for the last couple of hours. I don't think that's a good use of anybody's time. So what I want to do is actually pull out just one or two brief observations from each of this morning's speakers, and then actually take the second half of the time allotted to me to talk about a workshop we had a couple of months ago at NIH that was put together by the staff, and actually Cindy Davis was one of the co-organizers, and Andre Marriott was one of the featured speakers at that meeting. So you've already heard part of the meeting, but there was a lot that came out of that 2 days. So when I was in school 40 plus years ago, you know, the intestine, the large intestine, was simply there to absorb water, nothing else. You know, no connection to the inner organs of the body whatsoever. And I think only in the last decade we've learned that the microbiome is actually one of the most metabolically diverse and active organs in the body. And so pretty much everything that the physiologists taught me was wrong, out of date, etc. But coming to Cindy Davis, who started the morning, but one of the points Cindy made is we need to think about nutrient-nutrient interactions. That's really important. You know, I think we've been diverted in nutrient and disease research by focusing on saturated fats alone and heart disease. And we're finding out that maybe that's not the best advice. So every day we eat 40 plus nutrients. Just do the math on that. Think how many combinations we've got, plus the interactions with genotype, the large inter-individual variation that occurs in the microbiome. So clearly, one size does not fit all. Then Anne-Marie Singh talked about food allergy and the potential role of the microbiome, but her emphasis was on the scant evidence on the microbiome's effect in food allergies. And despite the fact that there are thousands of studies published now on the microbiome, there's really only rudimentary understanding of the role of those organisms in generating food allergic responses or even maturation of the immune system. And I think that really was the theme of the first three speakers introducing this topic, that our approach to the microbiome is kind of like the auto industry before Henry Ford invented the assembly line and started cranking out Model Ts and the internal combustion engine and a car with 4 wheels that had rubber tires and steering wheels became the de facto standard. You know, before that we had electric cars, we had steam-powered cars, we had cars with bicycle handles as steerage, we had boat tillers as steerage, we had 6 wheels on cars. You know, go to an old car museum and look at cars from the 1900s. They're really different, and this is really one of the problems in the microbiome field. I think it has the greatest potential for health, but I don't think we have any definitive answers to give our colleagues in the food industry out here of what they should be changing in the diet, and or what probiotic strains really are going to help people. Federico Rey talked about that in his talks about we're just scratching the surface of understanding the role of the microbiome in TMAO metabolism. And the question about the fish, and you know, fish can provide 50 times the TMA that you get from food sources of choline, and we don't, he fully admitted, we don't understand that complex interaction. So in the second set of talks, Andre Marriott started us off, and the one point I want to make that Andre talked about was bacterial translocation from the gut to adipose tissue or to lymph nodes surrounding the alimentary tract. I mean, that's been known for years, actually. John Rambeau and I published in early 1990s, late 1980s, in rat and hamster models about bacterial translocation of viable organisms into the lymph nodes, and the complete reversal of that by providing adequate fermentable dietary fiber. Andre was using direct observation with a fluorescent probe to see the organisms there. Then Maria Marco talked about the need for the right bugs being present at baseline. I think that's a key concept to see benefits of dietary intervention that people tend to ignore, and that relates to the question that was asked about pre-selecting subjects. And, you know, in theory, randomization is supposed to take care of all of that, but if every one of us has a unique microbiome signature, I don't think we can randomize half the country into that. So that's a real problem. And the question about Akkermansia, which Cindy asked, I think is really important, because we have Akkermansia in fecal samples that we're making hypotheses about, but really it's where it is in situ, in the mucin barrier, you know, a foot up in the colon. Maybe that's where the action is. And of course, with humans, there are technical difficulties in getting samples from the proper location. But it's something that I think we do need to think about some more. And I actually found it a little difficult to decide what I was going to talk about from Colin Hill's talk, because that was really impressive. I think everything he talked about, I just sat there and said, wow. But one of the points he made was about treatment of the pigs with salmonellosis and looking at the lactobacilli in the feces versus the ileum. And the point is that scientists are focused on the fecal microbiome as a proxy for the colonic microbiome. And I'm not sure we still know that's really true, but it's certainly not true as a proxy for the ileal microbiome. And even though we have logs fewer bacteria in the ileum than in the colon, we still have plenty of bacteria there that are very metabolically active, metabolizing a variety of substrates that are getting rapidly absorbed through the small intestinal mucosa. And the final point Colin made about bacteriophage, they greatly outnumber the bacteria that are in our colon. And they're getting short shrift. So, you know, they're sort of the Rodney Dangerfield of the microorganisms. They're not getting enough respect. So what I'd like to do now is turn to what's actually on the slides and discuss the workshop that we had in June at NIH. It was co-sponsored by NIH, the Agricultural Research Service, and ILSI North America's Microbiome Technical Committee. Ooh, and this works. Fantastic. So the reason for this workshop was our reading of papers, meaning the federal government staff who fund research and direct intramural research on this topic, and noticing that there's just not a lot of thought given to the diet in microbiome studies. You know, we just say there are people consuming a typical American diet. Well, what does that mean? And it's really problematic for trying to reproduce things. I mean, I had a really terrific graduate advisor who told me I had to write in my materials and methods enough information that somebody can reproduce whatever I was publishing. That's not possible from almost any paper you read today. And so it doesn't matter really if it's an in vitro model system, animal study, or a clinical intervention trial in volunteers. There's really minimal intervention information provided. Even though we know that diet is probably the strongest driver of alterations in at least the bacterial portion of the microbiome. And I'm going to use microbiota as the characteristic term, but it simply refers to the fact that most of the probes we have available are for bacteria, but we do need better probes for all the other creatures that have been discussed this morning that inhabit our intestines. And in fact, this is an issue for all microbiome studies. Earlier this week, I got an email that sort of looked like an announcement, but it was really just trying to sell me some new reagent for putting fecal samples into a solution that would preserve the DNA better than freezing. And I don't know if it worked or not. I've been traveling all week, so I didn't actually go and read the paper. So the idea behind this was to improve the rigor and reproducibility of federally funded research studies on the colonic microbiome and identify what information should be thought of in design of the studies and what needs to be reported. You'll see studies in animals that we fed Purina rat chow. Well, that doesn't mean anything to most people. In fact, you can't find out exactly what's in Purina rat chow because there's a computer in St. Louis that makes every batch of it depending on the nutrient composition of the various ingredients that go into Purina rat chow. There are lots of alternative diets, but they're often not identified in enough detail. And certainly for human studies, that's really problematic. So I'm still massaging all the comments I got from the 16 speakers who spoke in the 2 days at the workshop. And we're submitting the paper to the BMC journal Microbiome. So the workshop was divided into 4 sections, and the first one was on characteristics of dietary fiber and other nutrients that feed the gut microbiome. And the keynote talk in that section was from George Fay, University of Illinois. And George actually talked about plant structure and how cellulose in vascular tissue isn't metabolized nearly as well as cellulose in intervascular tissue. So the flat part of a leaf gets digested more than the ribs of the leaf, simply because of the accessibility to microbial enzymes in the gut. And of course, the types of fiber make a huge difference. You know, the food industry wants us to tell them what isolated purified fiber needs to be added as a prebiotic. I wish I could give that answer. We need much better information on how foods and food additives lead to shifts in microbial digestion. Benoit Chassaing from Georgia State gave a very nice talk about food additives, emulsifiers in particular, that have potentially significant effects on the microbiome. This is done at levels of consumption that are much higher in mouse studies that are much higher than what we would consume, but it certainly raises the question, what's the effect when we're consuming it at levels of ordinary exposure and combinations of those exposures, which we have really no information on. So then we moved into animal models. We talked a good bit about providing the reason for the animal models, and there's a lot of debate about should changes in the mouse microbiome dictate what we recommend for humans. You know, the mouse microbiome is substantially different. Should we only really rely on humanized mice or mice with defined microbiota? And there's no solid answer to that. Clearly, if you're interested in mechanisms or compounds that affect microbiome, if those bacteria are in both rodents and in humans, you're on somewhat safer ground. But I think it behooves scientists in the food industry to have confirmation from clinical studies on whatever comes out of animal models. Jean Chang talked a good bit about circadian rhythm in mice, and We need to know if this applies to humans. It's a lot more difficult to ask humans to provide a fecal sample every 3 hours. You know, that unless you've worked for David Jenkins in the University of Toronto, feeding the simian diet where you can produce a fecal sample upon request because you're eating 80 to 100 grams of fiber a day. And that's why David can only feed that diet for 3 weeks to people who work in his lab. Nobody else will do that. You just can't get the circadian rhythm nailed down in humans. And then, of course, Andre gave a beautiful talk about polyphenols modulating the microbiota in that section. Already mentioned Benoit's talk. And then Kelly Swanson from the University of Illinois gave the last talk on this section on non-rodent models, both dogs and swine, as potentially better than rodents because of differences in the anatomy and physiology of those larger animals. But none of those animals' models are validated as models for humans. We had a short section on in vitro models, a couple of talks there. And in fact, Devin Rose started it by asking people in the audience. There were 100 people in the room because that's all the seats we had in the room, and we had 120 plus online listening to this workshop. And Devin asked the folks in the room, how many people have collected fecal samples from donors where they simply reported they followed the American diet? And pretty much everybody's hand went up for that. Nobody ever asked how much fiber are these people eating, what fat are they eating, and so on. We had a talk about bioreactors, and given all of the limitations in vitro models, I think there's still a very significant role for them because you can study not only single organism effects, but you can study a defined ecosystem. And if all of us have a few hundred to potentially more than 1,000 organisms, it's almost inconceivable that, and we all have different signatures, it's almost inconceivable that we're going to have one set of standard bacteria for everybody. So we can obviously apply standardized treatments. It's great for screening an in vitro system before you invest in an animal study or a very expensive human study. And you can sample these things very easily. There are these small bioreactors, but there are also the multiple chain incubators like the SHIME system, where you can actually have 3 levels of the colon reproduced. You do need a steady state, and some of the systems reproduce that. Some of them don't necessarily ever come to steady state. There was a lot of discussion about pooling individual samples, and if you put 2 fecal samples together, there's always going to be one winner. But I don't think anybody has ever actually taken multiple samples, like 10 or 15 different individuals, and pooled them. One would think you get enough diversity there that maybe there won't be a clear single winner, but that you'll be getting potentially valuable single strains that are modulating the rest of the individuals there. And so all of these 3 portions of the workshop culminated in the final session on human studies. And you've already heard a little bit from some of the previous speakers today. We talked about the large differences in the microbiome of the Hadza, they're hunter-gatherers, or agrarian societies versus Western diets. You know, congratulations, you folks have the worst microbiome in the world. Not everybody's willing to switch to the diets that Venezuelan Indians and without any Western contact have. But some of our colleagues are actually experimenting on themselves right now. We need data on relationship of the gut along, of the microbiome along the gut and how that relates to the fecal microbiome, and more importantly, what that does to the plasma metabolome. Because as has been mentioned here, who's there is probably not as important as what they're doing. And one example that we talked about was fecal butyrate. You know, short-chain fatty acids are sort of the darling of people who want to analyze feces because nobody wants to work with feces, number one, and then you don't want to handle it a lot, so you just put a column for your short-chain fatty acids in your GLC, and it's easy to do. But, and again, what I learned was that the colonocytes use all the butyrate, doesn't get into the circulation. Well, that's nonsense. It's just not true. And in fact, there are G protein receptors in many distant organs besides the colon, identified in the liver, in the kidneys, in skeletal muscle, in the central nervous system. There wouldn't be receptors there if the substrate wasn't getting there. So we had one talk on fiber recommendations. In the United States, the dietary reference intake for dietary fiber is based on cardiovascular health. And Stephen O'Keefe basically said he didn't believe that. I'm from the federal government. I'm not allowed to criticize federal policy. I'm just going to say I agree with Stephen on this, that it seems to make perfect sense to me that the colon requirements should dictate what our fiber requirements are, not what our heart disease rate is. And clearly we need a lot more studies on effective range. Stephen's done some beautiful studies in the US and in Africa. His studies suggest we need 50 plus grams a day of dietary fiber. One of the questions we talked about and was addressed by Maria this morning was diet affect recovery of the gut microbiota after disruption with either antibiotics or infections. We had a number of other talks on studies with fiber mixtures. And in fact, it looks like fiber mixtures can be more effective than studying individual fibers if you're really disrupting the ecosystem using broad spectrum antibiotics. Controlled feeding studies are very useful. And in fact, most universities no longer have the capability of doing metabolic kitchen studies anymore. They're really expensive to maintain both the facilities and the staff to do that. Penn State has a terrific facility. The USDA, Human Nutrition Research Centers, all have those capabilities, but not a lot of other places. There's a handful across the country, but not nearly what there used to be 20 years ago. There are a lot of limitations of feeding studies because can't access tissues of interest. Mostly we're looking at intermediate outcomes. They're short-term. We're almost certainly not going to have long-term feeding studies with hard disease endpoints at the end. And of course, because of that, it challenges establishing cause and effect. We simply will be forced to live potentially with correlations. So we need better methods to determine dietary exposure besides asking people, what have you eaten for the last 2 days or 2 months or longer? So really, we have just a couple of general conclusions from the meeting. We need better reporting of design and control for both animal and human studies, and I would throw in vitro studies to that list as well. The attempts to characterize a core healthy microbiome and their metabolic functions still remains to be determined. So right now, I think the idea of a healthy microbiome is still marketing in a lot of ways. And it's, I'll just leave it at that conclusion. And so the bulk of scientists, I think, are taking the left turn here at the intersection. It says to the left, simple but wrong, to the right, complex but right. And we have a few people studying the complex but correct response to this. And I think I'm just about out of time, so I'll leave it at that. Thanks very much.
Well, thank you, Dr. Klurfeld. Are there questions? I know you're probably thinking lunch is out there, but this is a great opportunity to get some discussion going. There's been a lot of conferences for sure. I'll just throw something out. So it seemed like there was a fair amount of discussion at the NIH program on fiber, and yet we know that, for example, polyphenols may be acting to modify the microbiota. There's various things now that are non-carbohydrate based. And so I guess, do we need to have another session so that we can talk about more than fiber, or can you maybe just elaborate a little bit?
Well, because I only gave myself 10 minutes to discuss 2 days' worth of work, as I said, Andre presented his polyphenol work, then Marsha Singh talked about the emulsifiers. We had a number of presentations to talk about the potential of other ingredients that modify the microbiome. And in fact, I think polyphenols really deserve additional characterization as prebiotics in a lot of ways. So I agree with you completely. I think we, Justin Sonnenberg has coined the term microbiome accessible carbohydrates as sort of a replacement for both dietary fiber and prebiotics. But it doesn't cover some of the things Andre talked about this morning and Benoit has published on. We need to have a more comprehensive approach to this that you're suggesting.
David, thanks very much for a very entertaining and informative presentation. So science oftentimes leads regulatory activity. Could you speculate a bit on how the regulatory agencies may need to review current positions and moving forward as we gain more knowledge?
Nikki, you're going to get me in trouble with my boss. Dr. Parrish is from the Food and Drug Administration. I think the only thing I will say that probably won't get me in trouble is science is clearly moving away from one size fits all in nutrition, and in this area in particular. I think the FDA is going to have to actually look towards that horizon where people who carry these sets of bacteria might have to have one set of recommendations, and people who have that other set of bacteria might have to have another set of recommendations. But I think we're far away from you having to worry about that.
David, thanks again for this very nice summary, and this workshop that we had at the NIH that you organized was so important because it brought us together to discuss diets, as you said, in animals and humans. And one thing that we need to understand, I think is very important, and I had a few slides, but I skipped them because I was out of time. But I think a lot of people are criticizing that a mouse microbiome is not a human microbiome. But this is not only because these are different animals, it's also because we are feeding animals in our laboratories with diets that are not representative of what humans eat. And I think the most evident, the most obvious thing is the protein. Most people, including myself, using laboratory animals, the only source of protein we're using is casein. I don't know of any human only eating casein for months and months. So I think, when we're going to start using in the lab diets that looks more like what real people eat, maybe the animal and the mouse and the rat microbiome will start to resemble at least a little bit better the human microbiome. At least I would like to challenge the field to at least test this hypothesis, and maybe we're going to be able to say at some point that yes, the mouse is a good model. It's not a human, but it's a better model than we think for looking at a human microbiome.
I agree completely. I think one of the limitations there is just how much work's involved in preparing a diet that Andre's talking about, just freeze drying the components. We did that with meats, where we, graduate students, of course, in my lab, cooked meats, ground them up, lyophilized them, and the one student who had to do the beef didn't eat a hamburger again for a year and a half.
I just quickly wanted to add to what you had to say. I think it's a very novel thought. Does that not still, however, leave the challenge that at baseline a mouse model is pretty much identical, whereas what you said earlier is humans are all over the place?
That's a very good point. And I used to think that because we use animals, you know, these are basically brothers and sisters coming sometime from the same mother, that the microbiome should be quite homogeneous among the mouse that we use. But actually, even with the mouse that we use in our labs, we see quite interesting differences between the different individuals. And it seems to be related to their behaviors, the dominance between different animals living in the same cage, even the interactions with people taking care of these animals. Small details like wearing perfumes, for example, has an incredible impact on the gut microbiome. So there's a lot of little details. The devil is in the details, right? There's a lot of little details that even for a mouse model in a laboratory can have an impact on its baseline microbiota. And we need to understand this better. It shows you the complexity about studying the microbiome.
Okay, any other thoughts, questions, comments?